Okay, so thanks for inviting me to this discussion meeting on population genetics and evolution. Let me start by saying that I'm not really a biologist. Actually, I'm not at all a biologist. I'm a theoretical computer scientist uh, who's trying to look at problems, fundamental problems in evolution, in particular basic models, which people have looked at from a mathematical point of view, and trying to understand whether the results that have been proved in these or claimed to have been proved in these settings, to what extent are they true. And as in this talk, I'll tell you a little bit about what I have found, and as it will become apparent that many of the problems still remain open from a mathematical point of view. So the two models that I'll be talking about are the famous Eigen's quasi-species model. This is a deterministic model of evolution in some sense, in the holds in the infinite population setting. And here I will look at the question of the existence of error threshold, which is an important phenomena that this model seems to show. And this notion has found its use in drug design strategies, and I believe Nsheeth will probably talk a little bit more about it. The second model that I will look at is a Wright-Fisher type model with mutation and selection in the multiple locus setting. And here there are two questions that I'll look at. One is the convergence of this finite population stochastic model to the Eigen's quasi-species model as the population size goes to infinity. As well as I'll also look at the question of how much time it takes for the population to reach its steady state. These are both important questions, both from a biological perspective and also from a mathematical perspective. So let me start by saying some basic things. I guess the audience here doesn't need much motivation why we study models of evolution. Well, the goal is to capture important statistics and more importantly explain phenomena and make predictions. And in this talk, I'll focus on asexual evolution, which means that the models don't have recombination. And applications of these include enabling antiviral drug design strategies, which I'm not going to discuss. Let me start by basics. So we'll think of RNA, DNA as a string of length L. We will focus on the binary case or the two allele case, the 0-1 case. And in this setting, the sequence space is the set of all L-bit binary strings. Okay. So this is the setting. So we are looking for a population which lives on this space. Then comes the fitness landscape. To every such sequence or string, there is a fitness associated to it, which captures its ability to reproduce in a given environment or survive, howsoever you want to think about it. And this will be thought to be a number which will be, for the sake of convenience, an integer 1, 2, 3, or as large as you like. So this whole function, the fitness function, is collectively called a fitness landscape. And just as a side remark, that in computer science, functions like this, which are defined on the Boolean hypercube, which is what this object is, are called Boolean functions. So here is how it looks like. For instance, this is a sequence space for L equal to 3. And I have just jotted down some fitness numbers. So these black dots are these sequences 1, 1, 0, 1, 1, 1, and so on, and the numbers written them are their relative fitnesses. It will also be convenient to think of a matrix associated to this fitness function, which will be a diagonal matrix with rows and columns indexed by all sequences. And the only non-zero entries are at the diagonal, which are the fitness of that string. Okay. And if it helps to think, you may imagine that this string 0, 0, 0, or the all zero string is what is called the wild type or the master sequence. Okay, so let me describe the model of reproduction. So there is a parameter mu, which is what is called the mutation rate. So we imagine a string copying itself, and each bit of it is prone to error while during copying. This this is independent for each bit, and also the probability that the bit is not copied correctly is mu, and the probability that the bit is copied correctly is 1 minus mu. So for instance, the probability that this string 100 will produce 001 will be exactly mu square times 1 minus mu, because exactly 2 bits flip and 1 bit remains the same. Okay, so this is the model of copying. And so the word symmetric means that the probability of 1 going to 0 is the same as 0 going to 1. In general, you can consider all these problems in the non-symmetric setting. Independence, however, is a crucial assumption. So the quasi-species of model of Eigen can be described now as follows. It is an iterative process, as all evolutionary processes. It works over the sequence space. The fitnesses will remain fixed throughout the evolution process. So let's imagine these fitnesses as of now. These are given to you. So these are given to you, these fitnesses. And since the population is infinite, we only keep track of the fraction of population at each site. Okay, so we'll think of the total population as just one, and at each site we think of what is the fraction. So for instance, here the sequence 000 has currently population fraction 0.3, 0.1, 0.1, and so on. This adds up to 1. So we break the, while describing this model, we break it into 3 steps. The first step is the reproduction step, where essentially each string multiplies its fraction by its fitness. So for instance, here it was 0.3 and the fitness was 5. So the intermediate population for this particular string is 1.5. It is nothing but 0.3 times 5. Okay? We are just breaking down into 2 steps the process of reproduction and mutation. So what this step does is it increases the intermediate mass more than 1, as you would expect, because certain strings here have fitness more than 1. Then one can apply the mutation operator to it. So whatever mass is there on each of these strings, it kind of distributes its mass based on this mutation operator. So the way to think of it is that a unit mass of a string sigma 1, sigma 2, sigma 3 will result in exactly this much mass of a string tau 1, tau 2, tau 3. So what is this? This is the number of positions where tau and sigma differ, and this is the remaining positions where they don't differ. Okay, so mu to the power number of positions that they differ. This is really the probability of transiting from here to there. And finally comes in selection. The intermediate population mass was more than 1, and in this particular case it was 2.5. And the normalization happens, and we are back to the case where the population is 1. So these are the 3 steps of evolution in Eigen's model, and this keeps repeating again and again and again. What do you expect to happen? So because all the forces are present, selection and mutation, you don't expect one species to dominate. What happens is that this converges to a steady state, which is some distribution on this sequence space, and that is what Eigen called the quasi-species. Okay, it is a function of the mutation rate and the fitness landscape. So fitness landscape is really something very complex because these are really 2 to the n numbers. So mathematically, if we use x sub sigma t to denote the fraction of population equal to sigma at time t, then we write down the equation of dynamics in the following manner. So the total mass is 1. In the reproduction phase, this much fraction x tau t gets multiplied by its fitness, so it becomes this much in the intermediate population. Then the mutation step happens as I described, and then the selection, which is just normalizing the total mass to 1 unit. So this is the 3 steps that I described in the previous slide. So the equation, the dynamical equation is exactly this, that the quantity of sigma at time t plus 1 is equal to this quantity here, which takes into effect both selection and mutation, divided by the total intermediate population. So you can check that this is something which is equal. If I sum this x sigma t plus 1 over sigma, then this will be exactly equal to 1. So what we are interested in is where does this process converge, and this is most conveniently studied in terms of a matrix operator, where the vector x t plus 1, which just captures. So it's a vector indexed by all sequences, and it just captures the fraction of each sequence. This can be described very conveniently using a couple of matrices. So one is this matrix Q sub mu, which is defined here. This A matrix is the one that I described before. It is this diagonal matrix. And this was the population at time t. And this is just a normalization by the L1 norm of this vector. This is matrix times the vector at time t. So once I keep applying this again and again, what happens starting from an initial population x0, what happens? Where does this thing converge to? Any guess? If I have a vector and I apply a matrix again and again to that vector, where should that vector converge to? Yes. So it converges to an eigenvector of this matrix. Okay? And that, after normalization, will be what the quasi-species will be in this case. And understanding that eigenvector is the key to understanding Eigen's model. So it's a so in fact this is it not only converges to any eigenvector, it converges to the dominant eigenvector, the largest eigenvector. And so one may, so the key here is, or the goal here is to try to understand this eigenvector. It's really a very implicit object, because it is described, it is this matrix A, which has arbitrary entries in its diagonal, can really make it very difficult for us to understand what this right eigenvector can be. Forget writing it in closed form, right? But let's look at some limiting cases. So when mutation is very close to 0, or there is no mutation, you would expect that the eigenvector is localized on high fitness string. That's one extreme. When the mutation rate is very high, which is close to half, which means that the output population has no correlation with the input population at any stage, right? Then you would expect the distribution to be uniform over the sequence space. And this is what led them, Eigen and his co-workers, to empirically observe a critical mutation rate, mu, at which a transition happens. So some number between 0 and half, from where there is a sharp transition from the equilibrium distribution being localized on high fitness strings to something which is completely dispersed. And so here is one of these curves from one of his original papers. These curves are nothing but relative frequencies of different sequences, and the x-axis is the mutation rate. So as you're increasing the mutation rate, the concentration of high fitness strings is going down, and at around I don't know if you can read it clearly, at around 0.1 here in this curve, there is a transition. Okay, which means that essentially you can't tell which is there is no clearly dominant sequence in the steady state. So in fact, they even went ahead and postulated a very low value of error threshold, something about 1 by L. So L, remember, was the genome length. Okay, so something around that was postulated, and their simulations were largely restricted to what is called the single peak model, where there is one sequence with high fitness and all the others have fitness 1. And this is what they call the error threshold. It has, at that time, the goal was for him and his coworkers to try to explain the origins of life or how genetic information could have evolved from nothing. And more recently, it has found some applications in drug design strategies for RNA viruses and even retroviruses. So, however, to make even sense of this question or definition, one has to kind of be a bit more formal. So here is how we define the error threshold. So one thing to note is that you will never have, except at mu equal to half, the uniform distribution. So, and that is not really what they were trying to study. In some sense, we want to understand the smallest mutation rate at which the steady state distribution V mu comes within a specified error close to the uniform distribution. So U is the uniform distribution. Okay, this is how in mathematics you measure closeness of two probability distributions. So V mu is a probability distribution and U is a uniform distribution, and we look at their L1 distance or the statistical distance to measure how close they are. So once they come epsilon close, for the first time, we say that we define that to be the error threshold. So why is this useful from the drug design point of view? I don't want to talk too much about it, but just to point out that people did some experiments to find out that several viruses actually live close to their projected error threshold. And this led to this notion of mutagenic drugs, and the strategy was to increase the mutation rate to drive these viruses past their error threshold, where they would lose their genetic information. So here one has to be careful, because once you're trying to increase the mutation rate, if you bring it closer to the error threshold from below, you might actually increase the adaptability of the population. On the other hand, too much, increasing the mutation rate too much means that you are also kind of affecting the host, because these mutation, the way you will increase the mutation rate will probably also affect the host copying system and so on. So from a mathematical point of view, the question is that can the results postulated by Eigen and his coworkers and can be proved mathematically, okay, or rigorously? Yeah, of course they were observed in by simulations and so on and so forth. But can we prove a mathematical theorem here? And the answer is no. One can construct pathological landscapes, quite simple ones actually, where the critical mutation rate is not much less than half. Forget 1 over the length of the genome. So clearly some assumptions had to be made on the fitness landscape to be able to prove what Eigen and his coworkers had observed. And last year, this is exactly what I did. I looked into this problem and proved that for a very large class of fitness landscape, which includes all the biological relevant ones, the answer is confirms these postulates of Eigen and his coworkers. So here is the result. Let me define the class of fitness landscapes for which the result holds. So I call this a CK fitness bounded landscape. This is a generalization of a single peak landscape, where now instead of one, many sequences can have large fitnesses, but they have to be bounded by some number, which I will call C, and the rest have to be 1. Okay, so the existence of error threshold will depend both on K and C in this, but we have no restriction on the spatial structure of these strings. So these could be arbitrary. They don't have to be close in having distance and so on. And the result is that for these landscapes, the critical mutation rate scales like log CKL over L. So as you can see, if I increase C to be something like 2 to the L, then this result becomes meaningless, right? And in some sense, that is the reason why this behavior is not shown is not mathematically provable for every landscape. So however, it does prove the observed bounds for realistic landscape and does establish that many RNA viruses do live close to their mathematically correct error threshold. I don't want to spend more or less any time on proofs. I would be happy to discuss this with people who are more mathematically inclined. But as I said, the key here is to try to understand the largest or the dominant eigenvector of this matrix. And here we use some techniques which were developed in theoretical computer science, actually, with respect to these matrices. So these matrices somehow mysteriously appear in theoretical computer science when we are trying to deal with NP-hard problems. Okay, and quite well studied. And in particular, one can use Fourier analysis and so on to understand very rigorously and for all parameters of interest the eigenvector. Okay, so that's all I want to say about eigen model, and I can maybe take a brief moment here to if someone has any questions. Yeah. Mm-hmm. They could represent the basis, yeah. Or they could just represent alleles. I mean, it depends how you think of it. Yeah. Yes. 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 So yeah. So yeah. You're right. So the analysis will hold even in that case. It's if you notice here there is the sign is not equal to. This means that approximately, so up to a constant. And once you go into this AGCT thing and consider more general mutations, all this will do is just change this by a constant. But the asymptotics of, I think the key here to note is that if I assume C and K to be constant, which is what really happens in biology, you really don't expect fitnesses to be very, very large. I mean, in principle, they can be, right? This number can be as big as 2 to the square root L. Then if these numbers are small compared to L, or 2 to the L rather, then the scaling is indeed as 1 by L. And that is, I think, the key point here. Okay, so the questions that remain here actually are probably mostly ignored by biologists, but I think they're very important. So one is the following. So one of the ways to determine this error threshold is to run the following algorithm, which is, how do I calculate the error threshold? I start from the mutation rate mu equal to 0. I calculate the eigenvector. Then if it is close to uniform, then I'm done. Otherwise, I increase the mutation rate a little bit, and then repeat until for the first time I find that I'm close to the uniform distribution. Right? That's how you would do this. The calculation of V mu is just an eigenvector problem, so you will calculate V mu. Here. You can calculate the steady state and just check that whether you are close or not. The problem with this approach is that this assumes that once the equilibrium, once the population reaches close to uniform distribution for some mu, when you increase that mu, it cannot go back and reorganize itself. So it assumes a certain monotonicity in the behavior, that as I'm increasing the mutation rate starting from 0, it can never happen that once it reaches close to a uniform distribution or it disperses, genetic information disperses, somehow it cannot come back. And this is not at all an obvious question in mathematics. But this is the basis of assumption, and all simulations assume this question. And I don't know the answer. I mean, I can't do this mathematically as of yet either. The second question, which is quite important, again, from a biological point of view, is the convergence to V mu. How many iterations it takes before you are close enough to the steady state distribution. Now, this is not only just a mathematical curiosity, it is also useful in application of drugs, because you need to understand the time span. If you're really choosing to, you know, deploy drugs based on this strategy, you need to know how many generations or how much time you need to wait before you start seeing the, so maybe for the model that you have in hand, it's possible that the error threshold is low, but this time is very large, probably much larger than the human lifespan, right? In which case, it's not a great strategy. So here it can be shown that it is governed by the ratio of the second to the first eigenvalue of these two matrices. However, getting analytical bounds on this seems a hard problem. In particular here, one would expect that the number of iterations scales something like the length of the genome, not the more obvious thing, which is the size of the population, size of the sequence space, which is 2 to the L. But it's not clear how to prove this. Okay, so I'm going to move on to the finite population stochastic model. Just quickly summarize the positives of this model, which I've already said. The negatives are that the model assumes infinitely divisible population and is deterministic, while real populations are often small and lend themselves to substantial stochastic effects, and you observe things like genetic drift and so on. And for the general model, which involves multiple locuses and multiple alleles and so on and so forth, and includes selection and mutation, there has been no rigorous mathematical treatment as far as I know. And there have been many, many attempts, though. So let me describe the model. This, for people who know the Wright-Fisher model, you can think of this as the Wright-Fisher model with mutation and selection over multiple locuses. So the parameters are the length of the genome, the mutation rate, and the fitness landscape are as before. Now enters a new parameter, which is the size of the population. Okay, which will remain constant throughout the evolutionary process. Now now we really cannot keep track of fractions. We have to keep track of the number of sequences of type sigma at time t, and this is no longer a number. This is a random variable, which means that it takes, it could be different values with different probabilities. The population constraint does ensure that the total sum of all these random variables is a constant. It's a number which is equal to n at any time. And the model, again, I'm mimicking the 3 steps that I showed you in the Eigen's case, the reproduction, selection, and mutation steps. They can all be combined, or in any order, pretty much it works. So each string sigma now produces a sigma copies of itself. So at an intermediate stage, if I had n sigma t copies of sigma at time t, then in the intermediate stage, I will have these many copies of the string. Then there is the selection step, where n genomes are chosen at random without replacement. Okay, this maintains the population size. This is the selection process. And finally, the mutation step, where each selected genome now mutates randomly with per bit error rate being new. So now this is an honest to goodness mutation. This is not just transferring so many particles of this type to this type. This is a random process now. Okay, and I'll illustrate. I'll give an example in a second. And as I said, you can swap some of these steps. It doesn't really matter. But these are the 3 steps, and they keep repeating. And you see that the population now becomes a stochastic set in the state space. And this is almost the Wright-Fisher model with mutations and selections, except that there you do it with replacement, and here we are doing it without replacement. It doesn't really matter so much. But the more important thing to note is that unlike unlike Eigen's case, where the state space was 2 to the L, now the state space blows up to something like n to the L. Okay, and this is huge. I mean, even for simple numbers like 10,000, where the genome length is 10,000 and the population size is 10,000, this can be 10,000 to the 10,000, which is like 2 to the 10 2 to the 100,000, which is huge. Okay. So this is a huge Markov chain, a state space or a huge Markov chain. So here's an example for the single locus in the single locus setting. So here the sequence space just consists of 2 bits, 0 and 1. We assume that the fitness of this red or 0 is 2 and this is 1, and the population size is 5. So at a time t, we have 3 red particles and 2 black particles. The first step is deterministic, which is just that the red particles, each red particles give rise to 2 red particles. So right now there are 8 particles, okay, out of which 5 are selected without replacement and at random from this. So this is what introduces the selection bias as well. And then these things mutate. So for instance, each red ball can mutate to a black ball with probability mu, and so so this is a random process. And this is the population at time t plus 1, and this keeps on going. So now, this sample is moving over the state space of Markov chain. So this is the thing we want to track, okay? And as I said, so this is L equal to 1. This is simple, but in general, this becomes a very complex thing to keep track of, and the state space just is huge. So every question we asked in the when we looked at Eigen's model just increases in mathematical complexity quite a bit. So what are the questions that we ask? So one of the first questions which somehow biologists care about is when we talk about finite population models is do their predictions tend to that of the quasi-species model as n tends to infinity? Okay, I don't know why this is important, but probably historically because this is some model which people use. So any model that you have, its predictions somehow should go to this deterministic model as the population size tends to infinity. The second question, I think probably the most interesting question in this setting is the question of mixing time. So here it's really you see the state space is huge, and the mixing time, which is the time it takes for it to reach the steady state distribution, could be as big as the size of the state space, which was something like n to the L. It's astronomical. The evolution could not have happened if the mixing time was really so close to the state space. So it has to be somehow less, or we have to understand the circumstances under which the mixing time is small. So not only again, so it has both biological and simulation. So so if you run this stochastic process, you would not know when to stop, right? It's not as easy as computing an eigenvector. So all you are seeing are samples. How do you stop? How do you know when you have reached a steady state? Biologists, what they try to do is they track some simple parameters of this population, and when these parameters stabilize, they decide to stop, declare that this thing is reached a steady state. Any mathematician will tell you that this is just wrong, because this, all that means is that particular statistics has stabilized, not the population has not necessarily reached a steady state. So the only way to do this is to prove a theorem which establishes a bound on the mixing time in terms of the parameters, and then run the process till that much time. And then you don't even have to check. You don't have to check anything. You just stop at that time and just output that sample. Okay? So the key question here is, could the mixing time be logarithmic in the number of states? And also of interest is, how does the error threshold scale with n? You can define an error threshold in this setting as well. And how it scales with the population size is also of interest to certain biologists. So what do we do here? So we prove some theorems. I mean, which the first one with Dikshit here in the audience and student Srivastav, we proved in 2012 that indeed it is true that the predictions of this model do converge to the predictions of the quasi-species model as the population size tends to infinity. And this was the first to establish a rigorous convergence between a realistic finite population model and the quasi-species model. What about the mixing time? Our paper contains some preliminary results under some parameter settings where the process could be shown to converge logarithmically in the state space, but the parameter space was pretty restrictive. Okay. So let me just tell you, I don't know how much time do I have? 10 more minutes, okay. So let me just tell you a little bit about the mixing time for the single locus case and what the result that I proved was. So let me remind you that in the single locus case, we are looking at only two sequences, 0 and 1. And one string 0 has fitness A, which is some large number, and the other one has fitness 1. And we have the mutation rate as well. And this was the cartoon I showed you before. So what is the state space here? The state space here can be actually quite simply described. We just need to keep track of the number of red particles and the number of black particles. So let A denote the number of red particles and let B denote the number of black particles. And the constraint is that A plus B is equal to n, because the population is always tied to n, and that these numbers are non-negative. So when there is no selection, which means that A is equal to 1, okay, then this is one of the first graduate problems you do in Markov chains in probability theory to understand the mixing time. And that turns out to be log in the number of state space, log of n. Okay, so let's just look at, in this single locus setting, the state space is just n plus 1, right? Because the number of solutions to this A plus B equal to n when A and B are greater than or equal to 0 is exactly n plus 1. So the mixing time in this, when there is no selection, is indeed log n. And what we showed with Dixit and Srivastav, that the mixing time will remain log n under when there is selection and mutation, but they are kind of somewhat restricted. Okay. So some in the parameter range A times 1 minus 2 mu less than 1. So for instance, if mu is very small, which means something like 1 by L, which is what you want to look at, then this requires A to be I mean really close to 1, and it kind of brings you back to this setting. Okay, A cannot be very much bigger than 1 if the mutation rate is very close to 0, and that mutes select. So recently I've shown that in this particular, okay, so this result also had its analog in the more general setting, in the more, when L is bigger than 1. So recently I showed that for the single locus case, one can prove a mixing time of log n for all values of the selection parameter and the mutation rate. So no restrictions. Okay, there are some technical things, and again, I can talk about the proof if someone is interested in. Let me just conclude real quick, and then I'll take some questions. So in conclusion, we looked at, so one of the things I am interested in is looking at fundamental problems in theoretical biology from the lens of mathematics, or from a theoretical computer scientist point of view. And I looked at two models, the quasi-species model, and there we rigorously explained the ubiquitous error threshold phenomena. And open problems, many open problems, out of which the two that I think are probably the most challenging and relevant are monotonicity and the rate of convergence. Then we also looked at the stochastic finite population model, where we showed the convergence to the quasi-species model rigorously as the population size goes to infinity. And here again, several problems remain open. In particular, as I said, the most pressing problem, I think, is to establish a mixing time bound for multi-locus case. Okay, and here again monotonicity and I mean suitably defined versions of the monotonicity problem, or how does the error does the error critical error or the error threshold scale with the population like this. This is the quasi-species case, and this is the finite population case. Does this fails? It does go to the quasi-species prediction, but how does it go quantitatively? This is of interest. And one possibility is, which can be, which is open, is does it go like this in this? One can ask if these models can be rigorously analyzed even with recombination, and I haven't started looking at these things yet because I'm still, trying to think about these other problems. But this is a very important question. And more interested, I mean, being an outsider in this area, I'm very interested in listening to during the break or some other moment if people have problems that they would like to, me to look at or just have a discussion on. And thank you very much for your time. Red is exactly equal to the probability or the fraction of reds in the initial population. So let me just state it. So you started off with an initial population with, let's say, I reds and n minus I blacks. Okay? Yes, so in that particular case, what happens is what is called the selection sweep. So, yeah, those two results are known actually. So what is known rigorously based on what I have looked at is this right Fisher with nothing, no selection, no mutation, with selection, with mutation. These have been classically analyzed. I mean, you can open the book of Duret and he will do this. Okay? But the moment you go into selection and mutation, even the single locus case, they have to move to this diffusion theory approximations to this. Yes. That's what I'm trying to do. You mean to predict the fraction of populations and so on? No, that is not at all clear. What they do is they essentially take the population going to infinity limit, and the predictions are the same as what the predictions of the quasi-species model are. In some sense, they lose the whole point of being in a finite population setting. The moment you start applying this diffusion theory or this Fokker-Planck and so on, you lose the whole point that you are working in a finite population, and the results don't depend on that. So yeah, this is the first non-trivial case of Wright Fisher with selection and mutation for all parameter ranges. Yeah, thank you very much. Yeah.